Good morning, everyone. Good morning. It's such a pleasure to introduce our keynote speaker today, Dr. Ann Carpenter, Institute Scientist at the Broad Institute of MIT and Harvard. Ann is truly one of the pioneers in image-based profiling. Her work has completely changed how we think about and use cell imaging, showing that cell morphology can reveal deep connections between diseases, drugs, and genes. Her team's cell painting assay has become a cornerstone across the pharma industry and even inspired how new companies like Recursion and Cezong are made. She also leads major collaborations like Jump Cell Painting, Oasis, and the new Vista, which are generating incredible open data sets for the whole community. Her open source tool, Cell Profiler, is used by thousands of scientists around the world, and her impact has been recognized with awards like NSF Career, NIH Mira, as well as being named one of the top 100 AI leaders in drug discovery. I had the privilege of being mentored by Ann during my time at the Broad Institute. Her guidance, generosity, and passion for open science have had a big influence on how I think about research and collaboration. Today, she'll share how cell painting is accelerating drug discovery and how image-based profiling is opening new ways to understand biology at a scale. Please join me in welcoming Dr. Ann Carpenter.
Hey everyone, I'm so happy to be here with this community. I love seeing what's going on, all the advancements being made in image-based sciences with this group. Really just a pleasure to be here. The talk today, everything's public. You can get a PDF of the slides and I'll show that URL again at the end or take pictures of things, whatever you like. It's all open. So I want to tell you some of the work that's been going on in the Carpenter Singh lab. We also work really closely with Beth Simony. Many of you know she's also been involved in SBI2 for quite a while, and even organizing this conference, which is quite a lift. Thank you, Nassim, for all your work doing it this year as well. I want to just begin with AI and drug discovery. AI and drug discovery has been seen as a potential to sort of rescue the pharma industry. I spare you from showing Arum's law, that showing that the pharma industry is becoming decreasingly productive over time. And many are hoping that AI is going to make a difference here. And there are some signs of enthusiasm. I mean, not only is it the case that there are many companies that are starting sort of from scratch in using AI as the very foundation of their goals for biomedical discovery, but also it's the case that among the top 20 pharmas, they're also incorporating a lot of AI tools in their workflow these days. So there's a lot to be enthusiastic about. And I would say that AI is getting better at some tasks faster than others, and it's pretty directly related to what data we have available for training models. So for example, everybody knows DNA makes RNA, makes protein, and we have a lot of source data for these different tasks. I mean, in some ways, we know exactly which DNA nucleotides make which RNA nucleotides. That part's trivial, and we know which codons, which letter codes of the mRNA make which proteins. So those parts are just like very defined rules that biologists have discovered. But we also have data here to say, if the mRNA profile looks like this, what do we expect the protein profile to look like? Or if the genome looks like this, which proteins do we expect to be expressed? So we have a lot of these kinds of data, and that has made it easier to train models in that respect. And one that, of course, has received a lot of attention, especially since the Nobel Prize awarding, is going from protein sequence to protein structure. A real victory here, making it much easier to design certain kinds of biologic protein-based medicines. So really a lot of excitement about the potential here for AI to advance this. Now, after this step, things get ugly. So we can bridge DNA and RNA and protein to protein structure, but how do we go from protein structure to actually curing human diseases? Figuring out which proteins should we be going after, figuring out how to modulate the different functions within the cell. And the reason this is so messy, the reason this is so hard, is that we don't have simple rules that we can follow. We don't have enough direct data to train on, and we can't usually experiment on humans. We can't do interventional, perturbational experiments on whole organisms. So this is what is a really challenging step for the pharma community, and a place where we hope that cell phenotypes can make a difference, using cells, especially images of cells, as a mediator, as a proxy for those cases where we really are trying to link genome and transcriptome and proteome function to actual human outcomes. And of course, images are my favorite kind of cell phenotype, and that's what we'll be focusing on today. And the question is, in this, this is completely oversimplified. There are actually hundreds of steps along the way to get a medicine on the market. But can we speed up individual steps using AI? And not just AI, but also automation. The vendor hall here is full of all kinds of neat options for improving automation in the imaging world. There's also clever biotech like pooled genetic screening, which we'll talk about today, which make things make the data production side much better. And we heard some great examples, for example, from Tahoe yesterday. But AI as well. Can all of these new technologies help us speed up these individual steps in drug discovery, doing things that humans could do, but just faster and maybe a little bit better? But then the second question is, can we surpass human capabilities? Perhaps there are things that these systems can do better than we ever could, even if we had almost infinite time and energy to spend on them. So we'll look at some examples today. I think imaging is, because computer vision is so advanced, it's an advanced section of AI, we have really benefited from that in biology. And so it has become easier and easier to identify and measure phenotypes in images. I can still remember, like, it wasn't that many years ago when we were like, okay, deep learning looks promising, but there's not really anything that's so concretely useful that we really desperately have to be using it right now. Whereas now it is often the case that we have image-based biological problems where deep learning segmentation, for example, or tracking is really actually concretely useful. So we've gone through that shift in our field, and it's been really exciting to watch that develop over the past decade or so. I want to point out Beth Simone's lab is my sister lab at the Broad Imaging Platform, and she's leading now the Cell Profiler project, and as well as Piximi, which is a web browser-based deep learning focused phenotype classifier, and also doing all kinds of other image analysis tasks. Maybe the most important thing I could say today for those of you working on image analysis problems is, if you're ever stuck on anything, be sure to go check out image.sc. Image Scientific Community is what it stands for. And it is truly a community. This is a really friendly and welcoming scientific community, and if you don't believe me, go check out like how the genomics people treat each other. But there's just lots of welcoming people that if you're stuck on any problem in image analysis, there's really no excuse. You should post some images, post some kind of pipeline or workflow that you've worked out, not just for Cell Profiler, but for really any open image analysis problem, and people are often willing to get you along your way. And in part, our contributions to that have been funded by the NIH Center for Open Bioimage Analysis, which now Beth and Kevin Elisari are leading. So that's measuring known phenotypes. If you know what it is that you want to look for, and the whole field of high content screening is based on this principle of, we have some phenotype that represents some disease that we're really excited about, and we want to be able to measure it accurately so that we can score usually drugs or chemicals, potential candidate therapeutics, but also sometimes genetic screens to look for targets. And that image-based high-content screening, I would say, is really now a quite mature field. Most problems are quite solvable, and that's allowed us to carry out all kinds of great phenotypic screens. What I'll talk about for the rest of today is image-based profiling, where instead of going after one particular thing in images, which might require measuring many features in order to diagnose, here we are treating images more as an unbiased data source. We don't know what we're looking for, so we're capturing all the features and looking for patterns in that data. And here, of course, we use the cell painting assay primarily for this, and we'll be talking more about that today. I know many in this room, this is well understood, but I just want to make sure some are at this meeting because you want to learn more about this field. So I want to make sure we're all on the same page by just describing the basic high throughput image-based profiling setup, where we have these multi-well plates full of samples. We do the automated imaging using one of the imagers that you see in the exhibit hall. And we measure all kinds of things about the cells. And sometimes the things we measure are very concrete and biologically interpretable. So it's things like how many nuclei are there, how many speckles are there, how many synapses are in the image. Sometimes they're things like the shape and size of a particular compartment of the cell. Other times it relates to the intensity and texture of multiple stains. And usually you can get 4 or 5 stains pretty easily into an image-based assay, so there's a lot of rich information there that can be captured. But we don't always use software like Cell Profiler to measure those concrete engineered features. Sometimes we use deep learning feature extractors, and I don't show you any images of that because it's a bit more of a black box, where either individual cells, crops, or just patches of the image are randomly cropped in order to extract basic computer vision kinds of features out of those based on some kind of training. So I'll skip over all of that, how that is done and what the different options are there today. But suffice to say that at the end, either way, we end up with lots of features and lots of single cell data. And this is just, maybe 50 or 100 cells, but in reality, we often have millions to billions of cells in a given experiment. And what's so exciting about image-based profiling, and I'll skip the data, but if you want to email me, I'm happy to point you to other talks where I dive a little bit deeper on why imaging versus mRNA profiling. And the answer is that it is typically much cheaper, even for bulk profiling, but especially for the single cell level. All image data is typically produced at single cell resolution, and so just allows dramatically larger experiments, which of course allows dramatically larger discoveries. Because the predictive success of this data type is actually quite similar to mRNA profiling. We've done a number of experiments where they're kind of neck and neck, and often the data is complementary. So there's some discoveries you could make, like a gene compound matching, for example, that you could have found with mRNA profiles or with image profiles, but then others are unique to mRNA profiling or to image profiling. So very complementary data types, so we love using them together where possible and where we can afford it. So here's the cell painting assay. It's got these 5 channels with 6 dyes that are in them, because we double up this channel here. And it shows 8 different components of the cell that are every cell biologist's favorite organelles. We left out a few, couldn't fit them all in there, but this has been gradually improved over time to yield what we currently have been quite stably using. The version 3 of the assay has been pretty standard for a while now. So I thought I'd give this group might be a little bit interested in the history of cell painting. In 2003, I began writing Cell Profiler because I desperately needed it for my project as a biologist during my postdoc. And over time, I converted into a computational person. By the end of my postdoc, I was ready to start my lab at the Broad as a 100% computational lab, which was quite a shift. But it was a very exciting time to be in high content screening because there was not great software for measuring the kinds of phenotypes that are now very easy to do. The history of profiling specifically was really inspired by Zach Perlman's science paper out of Tim Mitchison and Steve Altshuler and Lonnie Wu's lab, where they did this multidimensional drug profiling by automated microscopy. So the image analysis segmentation algorithms were not great, so it was a little messy data at that time, but even so, they were able to group compounds by mechanism of action. And I found that absolutely inspiring, and it guided a lot of my postdoc work, such that by the time I gave my job talk at the Broad, I was pitching this idea, this future work. Hey, maybe we could do this large-scale image-based science, and we could group things like genes and compounds based on their impact on cells. Once I was at the Broad, Stuart Schreiber's lab brainstormed together with us, and here's some notes from that. Eventually, we decided to put together this assay with Sigrun Gustafsdottir leading the assay development to create cell painting. Let's find the cheapest possible dyes that measure the widest variety of things, and let's smash them into the channels as best we can so that they're still interpretable. We actually ruled out the idea of making cell graffiti. At some point, there was the idea of like, let's take 30 fluorescent dyes and just dump them on the cells, and like, who knows what's what, but it will make a visual fingerprint. Today, I think you could make such an assay and people would believe it and understand it and be like, okay, that's a cool way to get information out of images. But at the time, we were much more practical about the culture of biology, and we knew that if we told somebody, ooh, our magical assay system tells us that disease is differentiated from healthy controls, and then the biologists would say, ooh, cool, how are they different? What's different? And we would just have to say, well, our black box assay and our black box algorithms tell us that the images look different, but we can't tell you exactly why. We knew that would be pretty unsatisfying at the time. And then ultimately, I'm not a founder of Recursion. I'm on their scientific advisory board since the beginning, but that was the first company based on these concepts of image-based profiling, at least to my knowledge. So that's a little bit of the history, and for the rest of the talk, I just want to go through some of the different applications. I mean, this is kind of like a victory lap for the field because so many neat things have been done in image-based profiling over the past decades. So. The first one, I think, is one of the most profound kinds of things you can do with images, which is to say, can we use images to identify signatures of disease that we just didn't know were there before? Because if we can, then we can screen drugs to reverse the signature. We can also do genetic screens to figure out what targets might be involved in regulating the mechanisms of the disease that we care about, and so on. So we can learn more about the disease while also finding small molecules that might make a difference for patients. So the fundamental premise here is like, what if there is enough information in these images to tell us that this cell is diseased and something is wrong with some structure or some function within the cell, and structure and function being so tightly related in many cases. It won't always be the case that you can see using imaging, no matter what stains you use. Some will be invisible, but we wondered how often will it be the case that you can see what is wrong with a cell in a given disease? Because if we can see that, then we can treat with a bunch of different drugs and look for a case, this is just schematic, but look for a case where the drug is reversing the unhealthy looking disease state and turning it back into a healthy state. So very similar to what we heard yesterday about using Tahoe's data for mRNA profiling. This is the same concept, and other companies like Celerity were based from the beginning on this mRNA profile reversal, but now we're seeing, can we do this with images? And of course, there's some scientific and practical basis of images. Actually, structure really does tell us about function in very many cases. So for example, if a child looks a certain way, we can diagnose Down syndrome and also understand that the child probably also has organ issues, internal organ issues that are correlated with those structural changes, and likewise. So very many disorders do reflect in terms of their structure. Now, sometimes you have to zoom in a little bit, whether it's taking biopsies or blood samples to see that, okay, yeah, there is something wrong here. The whole field of pathology is based on there is something wrong with the structure here, and it is telling us something important about the human function. And so we're just really taking that concept, but we're doing everything in a very constrained, nice in vitro cell model where we can infinitely perturb cells with drugs and with genetic perturbations to try to figure out what is going on with those different disease-related phenotypes. So next up, I want to talk about how do we find phenotypes for every disease using images. So the idea of what we're going for here is, can we take cells that are in a healthy state, induce the disease state in some fashion? We'll talk about several ways to do this. What's shown here is knocking down a gene that is a known loss of function gene that causes a human disorder, cerebral cavernous malformation. So it is known that when humans lose function of this gene, they absolutely get this disorder. It's a monogenic, very simple genetic disorder. And clearly, a very obvious visual difference, and so now can we screen drugs that reverse them? This was the founding example for recursion. This work was done at the University of Utah, and they really scaled this up in quite a dramatic way, such that they now have hundreds of disease models that are available for screening in parallel, and many clinical trials underway. So making a genetic mutation is one way to induce a disorder in a dish. However, we don't always have monogenic diseases, and many of the larger diseases we care about tend to be genetically quite complex. And of course, obesity, metabolic disease is one of the examples where lots of genes are involved, but none of them are purely causative. And so here, Melina Klausnitzer took the clever approach of taking an array of patient samples, so cells taken from patients, where the patients had been genomically sequenced, and some of those patients had a very low risk of developing metabolic disease, and others had a very high risk of developing metabolic disease over time. And it didn't matter if the patients were currently obese or not, it was just a question of, does the genome of this patient lend itself towards having a problem with these particular symptoms? And so using this approach, they adapted cell painting slightly by swapping out some dyes and putting in a lipid dye. So these green fat droplets are very obvious to the eye. It turns out probably we didn't need to do that. They probably could have just used the adipocytes as they were and seen the effect of these fat droplets. They're pretty obvious in some of the other channels as well. But just to cover their bases, again, for biologists, it's typically very common that they really want to be sure that they're looking at what they think they're looking at. And so in the end, they've used this assay system to discover, yes, there is a distinct difference between low and high risk, and all kinds of other findings that are described here and more in these papers. So that's using patient cells that have a certain genomic precondition. For some disorders, psychiatric mental conditions are really challenging because there's not single gene causative mutations. It is also the case that we don't have great polygenic risk scores for them yet. And so in this experiment, which we just put on bioRxiv, we took patient cells. This is led by Bruce at McLean Hospital here, one of the premier mental health facilities in the region. And Donna McPhee together, they have painstakingly collected patient samples over many, many years, ultimately looking to see, for patients that experience psychosis, which includes bipolar, schizophrenia, and so on, do we have any morphological change? So growing these patient samples was super tedious, but they did manage to find a phenotype that's associated with psychosis. And because we've identified that phenotype, NIH NCATS adopted the assay and has run a 30,000 compound screen, which we're just now analyzing. That's Johan in the lab that is working on that data right now to see which compounds look like they might make the patients who experience psychosis look more like control patients. And here's the numerical data for that. Here's control patients, and the mitochondrial dispersion is what we're looking at. How far do the mitochondria extend away from the nucleus? Normally, mitochondria are relatively clustered at the nucleus, and they extend out to some degree. And here's what that looks like in control patients. Each dot here is a patient. And if we look at this whole set, these are all patients that experience psychosis in one way or the other. So here's the three individual categories of bipolar, schizophrenia, and schizoaffective disorder, and then the purple shows all three of those lumped together. And what's exciting is that, of course, we see that these are significantly different, and then what we see in major depression is that the patients look more like controls, and in fact, a little more in the opposite direction. Not significant here, but enough to make us feel really comfortable that this is a real phenotype that is associated with psychosis, and that gives us a lot of confidence in the drug screen that we're doing. With Ralda Name's lab, you heard her speak yesterday, hopefully, if you were here. She has adapted cell painting to neurons and many different types of brain cells, including astrocytes and neuroprogenitor cells, stem cells, and so on, and made all kinds of discoveries, which she described yesterday, and I'll skip here for today. And then the last example of how do we find phenotypes. So what have we covered? We've covered make a genetic perturbation if you know a gene that causes this disorder, look at patient cell lines with genomic risk if you've genomically sequenced them and you know a polygenic risk score that can predict the disorder, and then just take patient samples and just brute force patients with and without the disease, can we see a difference? Now, rare diseases present a really major unmet need. For those who aren't aware, they're not that rare overall cumulatively, but it's just not practical. Society can't afford to spend the money that it would take to cure 10,000 diseases. We're not doing very well with the hundreds or so that are very common. And so currently what you would do if you wanted to pick a disease and work on it is try to figure out some impaired function, or do cell painting on patient cell lines, as we just described, figure out some assay that is relevant to the disease, and then screen compounds. And this is estimated to cost $2 million per disease just to get a chemical hit that looks maybe promising for a given disorder. And then you've got all the rest of the stages of drug discovery, which are, as you are painfully aware, extremely expensive, averaging something like $1 billion per disease. So clearly we're not going to do 10,000 times 1 billion. So we have to do something different for rare diseases, and since we're in academia, we have a special opportunity to repurpose drugs because we don't care if it's commercially viable. We just want to ideally take an existing drug, find some phenotype, and then take some existing drug and see if it can reverse the phenotype. Existing drugs, of course, are cheaper in terms of development. We just have to connect it to the disease, and then it can be tested in humans. And so it's much cheaper, but also much, much quicker for the patients that are suffering from these disorders. They can take something off the shelf and give it a shot. So along these lines, for rare disease, how are we going to find these phenotypes? And the answer is a giant collaboration. There's dozens. I gave up trying to put all the names on the slide that are involved in this project because it's literally dozens across these 6 different laboratories. Even in my own lab, many people have been involved in this project over the years. The current lead is Runqi Shen, who hopefully you've met at this conference. And the goal here is to try to find phenotypes for screenable image-based phenotypes for many rare disorders highly in parallel. So the goal here is to predict the impact of genetic variants that have been associated or not with disease. And it's very useful to know this particular mutation at this particular location, does it cause a disorder or not, to guide the patients and the doctors to further research so we know what gene are we even looking at here, and to identify phenotypes for drug screening. So in this experiment, we are testing all kinds of variants. Some are pathogenic, some are known to be pathogenic, some are known to be benign. Others are a little confusing. Variants of unknown significance means we don't know. Conflicting means some sources of data indicate, yeah, they're probably causing the disorder, others indicate not. And so for these, we want this experiment to reveal to us, are these variants actually impacting some cell function or not? Are they pathogenic or not? And for the ones that we already know are pathogenic, we'd like to know why they're pathogenic. What are the functional impacts? And what are some screenable image-based phenotypes that we can actually use for screening drugs? So in this experiment, this whole experiment, the reason it's so large is that part of the team is working on measuring protein-protein interaction data, which is a super valuable data type to have, and others are measuring the abundance of the protein. So when that variant is present, which proteins interactions get messed up? Is the protein just not as much there in the cell? But what we'll focus on today are the image-based phenotypes of localization and morphology. And in standard cell painting, you might be familiar with those stains. By contrast, in variant painting, we drop a few of the channels so that we can use fluorescently tagged protein to figure out where is this protein localized. So this whole project is based on protein localization, but we also do, since we have these other channels, we also measure the morphology in a kind of more traditional cell painting kind of way. So the analysis for this involves using classifiers to tell us what's the difference between, hopefully control wells look like each other, And hopefully it's the case that we can distinguish when a gene has the normal version versus a variant that is known to cause a human disorder. So it's a classification problem where we try to build classifiers that can make these distinctions. And if you've done any amount of image-based data analysis, or really any data analysis from any high throughput screen, you know it's the case that you can build a classifier to distinguish any two samples. Even if I gave you an entire plate full of controls, you could build classifiers to distinguish any well from any other well. And that's because it's a beautiful sensitive data type that detects every possible technical artifact you could expose the plate to. And so we have to be a bit clever about setting thresholds and figuring out what is a distinction that is strong enough to count as a real phenotype as opposed to just this natural technical variation. And that's shown to some degree here. Here's the control wells looking like each other. They have a certain distribution of classification accuracy to be able to distinguish them. Sometimes you can distinguish control wells very strongly, and you can bet that some of those are on the edges of the plates as opposed to the interior, for example. So we define our threshold based on those control wells and then choose our hits based on those that are distinguishing the mutant form from the healthy form of the protein. And so this is what the data looks like for just a small set of these variants so far, just 1,000 variants across 100 or so genes. And we see it's actually quite common. So this one experiment that we are doing, which is not horrendously expensive, allows us to very rapidly test thousands of variants. And we're aiming to push to 20 or 30,000 of these over time. But for now, we can just look at this data here where focus on the orange pathogenic and the green benign. You see there's quite a difference in our ability to classify here, which means that pathogenic variants are more likely to have a mislocalization phenotype than benign. And of course, the unknown categories are in between. Protein mislocalization is pretty common. 14% of variants, 40% of genes is pretty exciting for a single assay to be able to reveal so many phenotypes. Altered cell morphology using the rest of the cell painting-like channels, much less so. The phenotypes are much less obvious than for protein localization. So there is a difference between pathogenic and benign, but it's much more subtle here, on the order of 2%, roughly tenfold less common. So we're focusing on protein localization for the most part, and Mikko Taipale was the instigator for many of these, for this entire track of looking at protein localization. He focuses on trafficking in his lab, and a single graduate student in his lab did the lab work to be able to test 3,500 human genetic variants and found 250 disease phenotypes in this project, and that's what we're scaling up in VERCHAMP to go even higher to increase that at least tenfold. Marzia Higigi and the group did the computational analysis for this experiment. And so the exciting thing is we already have 250 disease phenotypes discovered. Once we finish this, presumably we will have many, many hundreds, maybe over 1,000 different phenotypes. And we don't know any way to fund doing 1,000 drug screens. Even in the Broad, we do high throughput experiments at the Broad, but we're dependent on grants and collaborations, and we don't know how to fund 1,000 drug screens. So what are we going to do about this? The answer is that we're going to, first of all, make it a group effort, but secondly, we're going to be way more clever about how we accomplish this, because we can't just brute force do 1,000 drug screens one by one. And so coming back to this, now we're on the step of, okay, we've got 100 of these, 100 or 200 of these disease phenotypes. How do we screen drugs in a way that is reasonable and practical? And before I describe how we do that, I just want to provide some evidence that this is a worthwhile approach, that generally speaking, there are at least 2 drugs on the market right now that are based on correcting a localization problem. So for progeria, if you have progeria, the localization of the protein looks like this as well. The nuclear morphology is a little messed up. And when patients are treated with a drug that actually helps them, you see a partial reversal of this, not complete reversal even, just a partial reversal of the phenotype and of the morphology seems to help patients. And likewise for cystic fibrosis, for Vertex's drugs, plasma membrane is the normal localization. whereas that is disrupted in cystic fibrosis and, again, partially restored with the drug. So this is what we hope to find, but across hundreds to thousands of diseases at once. So in our pilot project, which, again, has a cast of many, many characters involved, J.T. Neal's lab is running the laboratory work, but heavily influenced by Paul Blaney's optical pooled screening. And here we use in situ sequencing to increase the throughput of the drug screening. So what we do is we take a 24-well plate, which probably looks a little silly to most of you that use 384 and 1536 all the time, but this is really a 24-well plate where each well gets one drug. And the reason the wells have to be so big is we are combining all of those healthy genes and disease variant genes that cause a protein localization phenotype. We're putting them all together. And the reason we can pool them all into one well is because each one is barcoded, has a genetic barcode that we can optically read out. So the readout of this experiment involves taking the images of the localization of each protein. Each one will have each protein has a different natural location and then a different location in the unhealthy state. And then the landmark other channels as well. And then we destain those reagents and then do in situ barcoding to read out AGGTC to tell us which of these different islands of cells got which gene, and is it the mutant version or is it the normal version of each of these different disease-causing variants. So that's how we plan to accomplish this technically. This is how we work around doing 1 or 200 separate drug screens. Instead, we pool it all. It's not as high throughput as an individual drug screen would be, but when you consider that it's 100 screens in one, it's really quite nice. Once we find potential drug disease hits in this screen, there's a number of steps that we'll take thereafter that are pretty efficient. First of all, of course, just do a confirmation screen and a dose response in the same cell type to make sure that we replicate the result in a non-pooled format. And then confirm in other cell lines that make sense for each disease. Here's where it diverges. Depending on what the diseases are that have nice hits, we can take cell lines that make sense for that disorder and see whether we still see these localization changes happening. And then from there, we will be engaging the rare disease community directly. There are a number of organizations whose very function exists to bridge this gap. So NCATS, Every Cure, and many other, Remedy for All, many groups that aim to take basic research in the lab and help translate it to clinical trials and to human patients. And so we will notify them, of course, with caveats, because this is coming straight from a cell-based experiment in the lab. But then we can brainstorm next steps. For drugs that are very mild and unconcerning, it might make sense to actually try them off-label in human patients. In other cases, there will be obvious lab experiments or animal experiments to do first to make sure that it looks like it will be effective. And what I'm excited about for this project is not just that we might actually cure some diseases within the scope of a not terribly expensive academic project, but also that we will identify many diseases that have shared mislocalization problems, which might incentivize pharma to take on some of these collections of diseases. So it might not be marketable to work on a disorder that affects 20,000 patients, but if it's 6 groups of 20,000 patients or more, then it might start to make more sense, and so on. So the scaling up that we're doing, we have a pilot grant, thanks to Anna Greca and Jillian Shaw at the Broad are working on this Ladders to Cures accelerator. So pilot grant has gotten us started. We are just now organizing the VISTA consortium. It's not started yet, and we'd be delighted to have more members join. So far we have 2, and the more we have, the more drugs we get to screen. And ultimately, I would love to see all of the drugs that have ever treated patients against all of the diseases that we are currently finding in Runshi's project, looking for new mislocalizations. So that's the long-term vision for this VISTA consortium and project. All right, so that was all of those examples we gave so far were how do we find a phenotype that's associated with a human disease. Next application we'll go to is can we identify gene functions by grouping similar genes and alleles with each other. So this project involved the team at Calico as well as several labs at the Broad Institute, and the basic idea actually uses the barcoding that I just described. So this allows us to take the whole genome CRISPR knockout reagents, pool them together, if I remember right, maybe 4 CRISPR reagents per gene separately, and we pull them all together with their genetic barcodes so that we can do here cell painting plus the barcoding and image analysis, and that allows us to cluster the morphological profiles. And of course, you can check the paper for all the neat things we did with this data, but this just gives you a little bit of a visual view on the genes that whose CRISPR did have a phenotype, of course a subset of the whole, and as we can see, they group very nicely based on their gene function. Now, this experiment was a bit underpowered. I forget, maybe it had like 50 or 100 cells per sample. Because of the barcoding, it's just the limit of the size. We could have done 4 times or 10 times bigger experiment, but that's the one limitation of this data set. It is publicly available, and we've gotten lots of neat things out of it. But I also want to turn to an alternate approach, which is generally more expensive and more challenging, but does give you a lot more cells per sample, and that's an arrayed cell painting project, which many of you have heard of, the JUMP Cell Painting Consortium data set. This was just really a delight. Like, despite COVID, despite everything, this group of researchers managed to get done a really amazing project. So collecting cell painting data following the same protocol at 12 different laboratories around the world. It got all these pharma companies up and running with cell painting internally, and was really just a heartwarming community project. A really great scale that now is giving us data that I'm sure we will be heavily using for at least a decade now, because it covers 3/4 of the genome are covered by either overexpression or CRISPR knockdown or both, and then we have more than 100,000 molecules plus the 30,000 from the Bray et al. Data set that the Broad made a decade ago, which we've reprocessed to be kind of in alignment with the rest of these. So tons of chemical data, tons of genetic data, just a really precious resource that I'm really excited to have. Single cell line, we had to make choices here that we'd rather have a lot more perturbations and just a single cell line, single time point, et cetera. But overall, 1.6 billion single cells are in this data set. And the data is available here. And just to give you a sense of scale, we heard about Tahoe yesterday. A lot of excitement in the virtual cell community about having access to 100 million. Oh, sorry, these are mixed up. 100 million cells. Previously, the biggest data set was collected by Chan Zuckerberg CZI. All of this is totaled up in a community crowdsourced repository called SC Base Count, apparently. So I have the impression this 300 million is like the sum total of all single cell data that exists in the world that is publicly shared and at least readily accessible enough to get into this repository. By contrast, here's JUMP Cell Painting, 1.6 billion cells. If you count the entire cell painting gallery, which has other experiments in it by now, it goes up to 2 billion. And the only other super big scale cell painting data set I know is RxRx3, which has 3.3 billion cells. And if we look at what data Recursion, this is made by Recursion, again, I'm on their scientific advisory board. If we look at what they have in their proprietary internally, it would like swallow this slide, because they have at this point hundreds of billions, maybe trillions of cells worth of data internally. So I raise this just because imaging is such a more efficient way to make single cell data, and it is just a beautiful kind of data, which again, I'm happy to share all the comparisons we've done to look at the information content. So I wish people would focus more on images than mRNA, but that's probably not news to anyone in this group. Okay, so looking at what can we get out of the data from JUMP Cell Painting, we have, for those who are wondering, when are you all going to publish the chemical data? The answer is we've been working really hard on it for a long time, and then we're coming to fruition of getting a manuscript out to describe the chemical side of the data. We did manage to get a paper out, thanks to Naranj, the postdoc who led this work, for the genetic perturbation side of JUMP Cell Painting. And so here's what the genetic perturbations look like in a snapshot. The majority of genes gave some kind of phenotype in cell painting, which is really remarkable if you think about this being a single stain set, a single time point, a single cell line. You still see a lot of changes that are induced by perturbing many of the genes in the genome. I wouldn't take I wouldn't say that overexpression is less sensitive than CRISPR, because these are not, we did not do the whole genome of both. We could only do 13,000 genes for overexpression because you can't overexpress genes that are too large, so there's some limit. We chose the 13,000 smaller genes, and the remaining 7,000 you really couldn't do even if you wanted to, because they just won't express well. So that's the limitation here, is that these are smaller genes. The limitation here, in theory, we could have done the whole genome with arrayed CRISPR. However, we used the druggable library from Horizon, and that, of course, is selected to be genes that are likely to be more involved in biological processes and pathways. So probably these are roughly equivalent, like you're likely to get the same proportion if we had tested a bunch of unknown genes alongside these guys as well. So that's how that looks, and that just tells you how many genes are doing something. How do we know the profiles are actually doing anything useful? And here's just one tidbit of data. You're welcome to check the paper for more, but this describes that if we, is it the case that genes that are in the same pathway as defined by WikiPathway, genes in the same pathway, do they look alike? Do they induce the same kind of morphology as each other? And you can see the results here. Here it does look like overexpression is a bit more fruitful than CRISPR. So that's the data set, and we're starting to have some lovely tools out there. So of course you can go to where we share the data is on Amazon Web Services through their ROTA program. Very grateful to AWS for sponsoring the data to be stored and shared for free. That's huge for an academic organization. I don't know how we'd pay for it otherwise. But we also building, so that you can just download the whole set or crunch on it on AWS as you like. But the other thing you can do, if you're just interested in a single gene or a single chemical, Alon Gonzalez in the group has built a really beautiful interactive set of tools that allow you to answer questions like, when my gene is knocked down or overexpressed, how do the cells look? What are their distinctive features? Which other genes look like my gene of interest? And so on, and likewise for chemicals as well. So those tools are available at that URL shown. And all this data is stored in the cell painting gallery. Again, that's on AWS, thanks to their ROTA program. And many thanks to Aaron Weisbart and Ank Kumar for getting everything in there and structured in a nice way so that the community can make use of the data we've collected so far, but all future data will be in this very nice format, so it's available to the community. All right. Moving on to a next application. So that section was all about looking for similarities among genes. Now the question is, if we find a phenotype associated with a genetic perturbation, can we identify chemical regulators of those? Ideally, without having to physically do experiments. So we talked about genetic pooling to allow us to do lots of drug screens in parallel at a much cheaper kind of way, but can we do it in silico so we don't have to do any experiments? And that's what we attempted here in this project, which was a collaboration with Karen Eisinger's lab. And for some reason, I don't have, Mohammed Rahman was the lead on this on our side. And here we take samples that are unperturbed and with controls, and then we overexpress or CRISPR knockout genes, and we look for a difference. And here the difference is super subtle. I don't really expect you to see it, but the computer can see it, and the question is, have we ever seen any chemical perturbations that match that phenotype? So this was kind of one of the purposes of making the giant jump cell painting data set, is to make a resource that people could do matching exercises like this. So this is an early project before JUMP was finished, so we used just the 30,000 Bray et al. Data set from the Broad from 10 years ago, but we did find matches across a number of different projects, and the paper describes 7 different kinds of genes that we went after, but here's just one of them, which I mentioned because the results of it were so promising. We found compounds that impact the hippo pathway and impact sarcoma cell growth that were so exciting that Dr. Karen Eisinger is now full-time, instead of professor at Penn, she is now full-time CEO of this startup that we launched to pursue these compounds, but also a platform that can find novel targets for solid tumor treatment, which are solid tumors that are otherwise quite devastating. And then I think the last story here is, can we predict the outcome of an assay by correlating its readout to cell painting profiles? And here I just want to give a cautionary note. I mean, the answer is yes, we can, and there are a number of different papers in the community that describe this basic strategy, saying, if I see a cell painting profile like this, it means that that compound that treated those cells, or genetic perturbation, is likely to also be positive in some totally different assay. As long as I train with enough examples, I can do that prediction. That's the basic concept. I just want to give a cautionary note from more recent work where the assay prediction benchmarks that are out there are too easy. So one of which we produced, Moshkov here, but then others coming from public PubChem data. And what I want to point out is that in each of these benchmark data sets, the task, the machine learning task, is can I distinguish compounds that are active from compounds that are inactive? And it turns out that cell count is confounded with activity in each of these studies, such that you can just draw a straight line. If a compound reduces cell count by some amount, it is much more likely to be an active compound than an inactive one. And that, of course, is not what people set up these benchmarks to be able to do. And so it can be very confusing to look at the results, because what you find is if you just try to predict which compounds are going to be active in assay 1, assay 2, assay 3, assay 4, you will do very well, this blue circle, just using cell count as the predictor, which of course is not actually biologically super meaningful. It just means that the benchmark is designed badly because it has far too many. What we need is to fill in this. We need a bunch of decoy inactive compounds in the assay, and yet they do something to the cells. They are somewhat cytotoxic to the cells. So we need to fill this in for these benchmarks to be really meaningful. And unfortunately, none of the benchmarks that exist are doing that. The one that is presented in the paper that is going in that direction, it has just a handful of assays that do meet that criterion. And the descriptions here, I don't want anyone coming away saying, oh gosh, cell painting is useless for assay prediction. That's not the takeaway message here. The point is, because the way the benchmarks are designed, cell count is too effective, and then it's hard to see any benefit by adding actual rich information about what the cells are experiencing with each drug treatment. And then this is a subset of assay prediction, is predicting toxicity. I don't think for this audience I need to explain why it's so important to predict toxicity. Most of you care about the pharma side, but there's also the agrochemical and commercial compound. 300,000 compounds are used in commercial products and have never been assessed for safety, which is, I find, actually pretty horrifying. And of course, on the pharma side, there's increasing interest to use new approach methods. These are non-animal methods in trying to figure out toxicity, largely because the animal models are not so accurate. And so we launched the OASIS Consortium about 2 years ago. David Rookie is the other leader of this project from Bayer. And the goal is to test cell painting together with parallel transcriptomics and proteomics data. So this will be just a beautiful data set. It'll be released July 1st next year with all 3 of these data types in parallel. Not a complete set because transcriptomics and proteomics are so expensive, but at least a subset will be available. And the goal to start with is predicting liver tox. If this looks effective, then we can move on to other kinds of toxicity as well. So here's what that consortium looks like. I'll skip the details here, but it's including basically every compound for which we have rat and human liver tox data, like whole organism data. It's a very small data set for those of you who are coming from the more machine learning side. It's a depressingly small number, but we're increasing it with some compounds that are donated from the industry partners pretty substantially. And then we're testing, each compound is tested against a whole series of different cell models, a subset on these more expensive ones, thanks to Johnny Sexton, who's going to be doing a ton of liver organoid work on these compounds and many more. And then all those different cell models, and then all these 3 multi-omic outcomes that I described, thanks to the collaborators, BioSpider, and I'm sorry I didn't add this here, Alethea Genomics is now doing some transcriptomics as well, and Nomic Bio on the proteomics side. And then we do data analysis and produce the data to share with the community. And here's just one little tidbit of that data in collaboration with Axiom, who did the primary human hepatocyte arm of this project. And I'll leave you to the preprint to look at the details here. But what we found is that cell painting was more sensitive to detecting toxic compounds as compared to some alternative strategies. Again, using cell count as a baseline here to make sure that we weren't, that the task is not too easy. And indeed, we find more sensitive to compounds, and we see changes in profiles at a lower concentration using cell painting. So I hope you enjoyed this little tour. These are all the things you can do with image-based profiling, and the green ones are all the ones we skipped today. So there's all kinds of other things that can be done. Mechanism of action determination is, of course, one of the biggest things that it's currently being used for in pharma, but it's kind of routine now, so I didn't emphasize that one today. But all kinds of different things here, different conferences and review papers that might be helpful if you're just getting going in this field. So thank you so much for your attention, and really happy to be here and answer questions.
Great talk, thank you. I'm really interested in the optical pooled screening. I'm wondering if, like, after you've collected the disease phenotypes for the 200 or so different diseases you have, can you use that then to barcode the cells rather than reading out a hybridization? Because in theory, each disease type has its own profile.
Oh, it would be impossible because there's a lot of shared mechanisms. So when we say localization change, there's probably like 20 categories of localization. Like some are ER to cytoplasm, some are cytoplasm, ER to Golgi, some are plasma membrane to cytoplasm, right? So there's only so many pairs of those. And if we just tried to use the localization, we wouldn't know in the presence of a drug, is it in the ER because it's normally in the ER, or is it in the, has it changed because of the drug? So unfortunately, yeah, we really do have to do the genetic barcoding to be able to figure that out.
Okay,
Yeah.
Thank you.
Hi, thanks so much for the talk. I'm not directly in the field, so I'm probably way behind the news, but I seem to remember seeing some publications a while ago that suggested that bright field images or label-free images have actually a lot more information because you get anything that scatters light and not just the 5 or 6 markers you're staining for. So I'm just wondering, can you tell us a little bit about like whether you're exploring just bright field imaging, label-free imaging, and where the field is at in that area.
You've hit on one of the more exciting things. Recursion has announced publicly that they are shifting towards bright field readouts because they can get a lot of the same information from just a bright field as compared to the dye-based assay. Cell painting's super cheap, but it's not zero to buy the dyes. So that's a big advancement that they've announced. And then on the academic side, we've seen a number of papers. I don't think we've yet seen a super convincing paper that has a large enough set of compounds or a big enough task to be really confident that bright field imaging is replacing 90%. Of course, we don't expect 100%, but is it even like 80 or 90%? We've seen some smaller scale experiments that look pretty promising, but I would hesitate to be conclusive, like, we don't need the dyes anymore, we can just use bright field. Part of the hesitation of switching entirely to bright field, so I should say, jump cell painting, a good proportion of the partners were able to include bright field, so maybe half of the sources or something, half of the data will have a bright field alongside, usually a 3-stack Z-stack is provided. So that data exists for people who want to play around with exactly this question. But we see, I think that there's the key challenge is how do you train the model to get as much out of bright field as you would like. One way to train it is to give it a bunch of examples of bright field plus fluorescent stains, not just cell painting, but maybe all kinds of other stains, and ask it to predict those stains and then extract features from all of those. So that's like a virtual staining kind of approach. That's one strategy to get data out. The other is to just use the bright field only by itself and only train on some kind of like semi-supervised, weakly supervised approach. And so not everybody can just do that. Not everybody has the data sets that are appropriate to do that training. So in my perfect world, in a few years from now, somebody has trained beautiful model that if you feed it a bright field image, it can not only produce all these other channels that realistically, but it can also extract a feature profile that is just as powerful. And I think it's possible, but it would really require, the problem is generalization. Like you can definitely do that for a given data set, like within one company that collects images consistently, you can certainly do that. But something that works for any lab, any of us that just produces bright field images, it's not there yet.
Thank you.
Hi, Anne. Thank you. Thank you for this wonderful talk. So I think, so maybe I'm in the side of the audience who are working on more like, well, we have a biological question and we are trying to apply cell painting, and then we go to present in our department and the cell biologist in the room always raises a hand. It's like, okay, you've painted, you get these features, so what? Mm-hmm. And so, you know, so sometimes I struggle with that answer of the bat because I can start wobbling around. Well, it's a manifold. We have many features. It's more complex than you think. There is kind of but how we make sense? What are the steps to make biologically meaningful intuitions about the feature space that we observe, like in a disease state, like, you know.
So it sounds like your department is not ready for cell graffiti, even now. No. But I would point you to, there's a blog post we wrote that's called "Help!" Interpreting image-based profiles. And sadly, it's a few years old, but it's actually sadly still pretty up to date for how do you make sense of the profiles. So like I see a cluster of genes, or I see a difference between disease and healthy. What exactly is changing? That blog post kind of talks you through some of, from simple, if you're lucky, then it's simple. And if you're not lucky, it's hopeless. So it takes you through that emotional spectrum. And it includes things like, step one, if you've used Cell Profiler for feature extraction, what are the names of the features, and do they group by channel? Do they group by style? Is it all size and shape, or is it all texture of a certain channel? So that's the simple end of the spectrum, and then it gets harder from there.
Right. I guess the challenge is when you see a manifold of features, right? So all of the above describes a phenotype, and we have nuclear features. We have also correlation of ER with mitose and something else. And so how to, I guess the challenge is like, okay, this is the complexity. This is the phenomenon. This is the phenotype.
Yeah.
So if it's manifold, how we articulate that in a more kind of intuitive fashion.
When it's a mixture of features across channels, across types, then it gets complicated. Sometimes if you ask me or Beth Simony, we've seen enough of these to be like, oh, that sounds like a really complicated mess, but actually it's probably mitotic cells, or something like some of them we've seen before, but I would say, a lot of them you hit a road wall. Now the good news is that the computer vision world is excited about this problem for all kinds of other reasons, selling ads and who knows what else. And so there are deep learning based methodologies that can kind of try to that show you what the model is paying attention to, that can be helpful, but they're not so amazingly successful that we do this for every project. We personally do. Most of the time we try to live without the interpretation. We try to rule out technical artifacts and then just move on with our lives. But if it's not one of the simpler ends of the spectrum, but there are methodologies that are a little painful to try to get at this interpretation question.
Cool, thanks.
Yeah. Well, thank you very much.